Okay, so I just tried to pull together a few slides so we can go home with the main messages in our mind. It's quite easy sometimes, isn't it? You sit after 2 days and your head's just so full with everything swimming around. So I've attempted to order it. So I have to apologize to all the speakers for summarizing your talks in one line, which I've quite possibly got wrong anyway, but I'll try my best. So we started off the meeting yesterday with, well, how do we actually treat patients who've got antimicrobial resistant bacteria? So you had Aidan Clokie talking about, well, the insights from license that give us these bigger host ranges. But then we had the sort of real life Mariam Dadiani talking about how she treats her patients in the Eliava Institute. So talking about the fact that she's at the front end and what she uses, first of all, her general phage cocktails, and then getting more specific. And she fully admitted they don't really know very often what's happening mechanistically, why it is the oral dosages seem to give efficacy. But nonetheless, still, you know, it's a different set of practical problems that she's trying to solve. Then we had Heather, who said, well, don't forget the bacteria that are the phages that are in the bacteria that you're treating. How are they communicating to each other? And so how are the prophages going to play into how we use bacteriophages therapeutically? And then we had Antonia summarized how we can essentially how you would look at engineering to benefit phages. But I think very often that people try, and it's really fun to play with phages and make them do better things, but they have been at it for a considerable number of years, 3.9 billion years. So I think it's definitely advantageous for us to do that, but to actually better understand the biology and then integrate that into our engineering. So that's what she was talking about. Then I think the next theme is just the sheer complexity of the biology. So I think I certainly noticed there's been a shift in interest from people not being very interested in using phages to all our local doctors, for example, are like, give us your phages, my patient's dying. But I think there's a massive chunk of science that has not been done because it's not been a very fashionable area to research in. So, and I thought that was I really liked the session we had, the second half of yesterday. I thought it was a really nice flow with Alison Lowe talking about, well, how could you make better predictions? And then Janine talking about, well, what about, what are all the different ways that bacteria generate resistance? That was a lovely theme this morning as well. And then Sabrina talking about, well, actually, even though you can see theoretically they can do all those things, what are they actually doing in the clinic? And then Pablo talked about we finally talked about the fact that the phages prefer the rough over the smooth, and how can we can actually encourage them to overcome this. We saw different ways of Theo from our lab trying to understand, at least even if we don't know what genes encode, we can get information from trying to look at their interactions in more detail. And clearly, getting these animal models is going to be really key as well to then using them in humans. Then I thought there was a really nice session in terms of tools. What's new in terms of all the tools? Talked about, there was a very nice couple of talks from Peter Bowen, either using the receptor binder proteins or labeling whole phages to make much more sensitive diagnostics. I think as we go forward and phages are used therapeutically, you're going to need that much better resolution of therapeutic information. We had a nice talk from Dorian talking about how you can essentially Legoize the license and arrange them in different orders to improve the specificities, and then different tools on how to nice talk from Rita showing, well, actually, if you just blast phages with some heat and you can see what survives and then use that to then figure out what to remove. And talked about, and there was a nice talk looking at using nanoporter sequence classes of genomes within the gut to understand their strategies. Then I thought we had this nice session really on how we choose, how do we choose all the phages that exist, how do we actually go about it? So we had these nice talk, Christian gave a nice talk from Exeter saying from that large library, how do you nail the breadth and the depth to get it right? How do you understand with the complement? I talked from Livy about then, okay, the translation, how do you make that happen within a legal context? And I thought Paul's parallels with HIV was really interesting. I'm sure there's lots of parallels from many other things we can bring in. I tried to summarize Aidan's closing. I think it's, even if you're blind and have to carry your harp and a donkey, you can still write beautiful tunes. So I think Aidan was teaching us about resilience. I don't know what I'll have to say about that. Then to today, we had, I thought this morning was excellent. I really enjoyed the whole string of talks from Jose's group, and it reminded me of something Jose said a few years ago when I heard him. I had him talking at a conference, and he was saying, phages and bacteria, they're not fighting, they're dancing. So here's a, not sure if these ones are fighting or dancing, but they have their boxing gloves, so maybe it's a bit of both. But essentially there is that, isn't that, there's that sort of the beautiful choreographic dances of it. They want something's about something of a counter, and then there's that sort of trying to pull apart all those things. I like that the very nice suggestion today on, they can stop the tails forming, they can mobilize all the defense systems across the place and target just the phages that have got plasmids, that of course the plasmids play into it. Then we had to talk about, well, how do we, that's sort of moving tack a little bit with Maria's nice talk as well, then how do we give our phages to our offspring, and how do we understand those dynamics happening? We had a nice talk on the Dia's talk on how we're actually using a transposon mutagenesis to identify again which genes phages are tagging and targeting. And then, of course, the methylation is another whole complication of phages, methylating things left, right, and center, and then the whole host ranges can be changed due to that. We just have problems in the lab because overly methylated phages clogging the nanocell. so methylation seems to be an intriguing, frustrating area of phage research. And then we had the nice talk on the arbitrium system, how peptides feed in. So it's sort of, I think if you can imagine the way that phages might use some kind of to interact with each other, they probably are phages that are doing it out there. So that was a good session on all of them. And then food and milk. So we just finished the session today, circling back to other places that we might want to use our phages. So how the nice talk on the taleosins, can we actually use them, not the phages? Can they change the mastitis landscape? Use them directly in food and to treat the animals in which we eat. So I think that was the sort of main themes. I thought hung together really well. Thank you for organizing. You said in your thing that you hadn't put them in a very good order, but it was brilliant. So yeah, with that, just thank you all for all the lively discussion and for making the conference so much fun to be at. So thank you very much.